Hey everybody, it's Mrs. Kraus, and in this video we're going to be learning about meiosis, which is the type of cell division that involves the creation of sex cells like eggs and sperm. So before we start talking about meiosis, I want to do a quick review of mitosis and the overall goal of mitosis. So you have a question in your notes, and that's right here, that asks, a human body cell has 46 chromosomes. And in a human body cell, mitosis is going to make, and you need to select the answer choice that has the correct number of daughter cells created and the correct number of chromosomes in each daughter cell. You're then going to provide an explanation below for your answer choice. Okay? So now would be a good time to pause the video and then come back to it once you've done that. Okay. So the correct answer choice was 2 cells with 46 chromosomes each. A key thing about mitosis is that it creates daughter cells with the same number of chromosomes as the parent cell. So if the parent cell has 46, the daughter cells also have 46. In mitosis, 1 cell divides to create 2 daughter cells. And if you want to provide an explanation for how we end up with 46 chromosomes in each daughter cell, you might say something along the lines of when the chromatids are split during anaphase, they are then considered full daughter chromosomes. We also did an assignment called the mitosis and chromosome number assignment, and if you look over that, that might help you understand better as well. Right below your explanation, it tells us that mitosis involves taking a parent body cell, like a blood cell, a bone cell, a skin cell, and dividing it to make 2 daughter body cells. Okay. Something that we want to keep in mind is that our new daughter cells are considered diploid, just like the parent cell. Number 1 on the right-hand side asks, what does it mean to be a diploid cell? For your answer to number 1, you're going to write new daughter cells are diploid, so that means they have 2 sets of chromosomes, which is 46 total in humans. For humans, each set of chromosomes is 23, so 2 sets would be 46. The name diploid actually makes sense because di means 2, so 2 sets of chromosomes. Then we have a little chart, both on the PowerPoint and on your notes, that compares diploid cells and haploid cells. We only make diploid cells in mitosis. Again, we've got 2 sets of chromosomes, and in humans that's 46, and we're making body cells. We only have 2 types of haploid cells, and haploid cells have 1 set of chromosomes, so in humans that's 23. The 2 types of cells that are haploid are sperm cells in males and egg cells in females. Both of those are sex cells. Okay. The next type of cell division that we're going to discuss is called meiosis. And during meiosis, we're creating these new sex cells, eggs and sperm, for the purpose of sexual reproduction. This image right here shows tiny little sperm trying to fertilize a single egg, and the egg is always much, much larger than the sperm. And what I mean by fertilization is the sperm joins with the egg, and they each bring 23 chromosomes to create a fertilized egg, which is called a zygote, that has 46 chromosomes. That zygote can then divide many, many times by mitosis to create a baby that has body cells with 46 chromosomes each. And this is our answer to question number 2, how does meiosis make fertilization possible? Okay, so we can say, hey, it makes eggs and sperm. They've got 23 chromosomes. When they come together, they make a zygote with 46. And that eventually makes a multicellular baby with 46 chromosomes in each of its cells. Okay. We have two types of reproduction. Asexual reproduction, which we do not use, creates children that are identical to the parent, and there's only one parent in asexual reproduction. An example might be binary fission in bacteria, which we talked about with our cell cycle and mitosis notes. And during binary fission, a cell, usually a bacteria cell, is going to make a copy of its single circular chromosome, and it's going to split down the middle. Another form of asexual reproduction is called budding. This happens in sponges and this little guy to the right, which is called a hydra. And what we see is that mitosis is creating a baby bud that's growing off of the adult. And mitosis will make more and more body cells for this baby bud that are identical to the parent cells. And eventually it gets so big that it pops off and becomes a clone of the parent. This doesn't happen in humans, obviously. Sexual reproduction is what humans use, and that makes offspring that are different from either parent, since there are two parents and they're contributing half the DNA each. So the offspring isn't identical to either parent. In order to create these sex cells, meiosis takes place in the testes in males and the ovaries in females, and then the sperm and egg are going to join during fertilization. Okay, now you have a question number 3, I believe, on the right-hand side of your notes that asks, what are the two ways in which asexual reproduction is different from sexual reproduction? So for the answer to that question, you should have something along the lines of asexual reproduction involves one parent and sexual reproduction involves two. So that's one major difference, is the number of parents. The other major difference is asexual reproduction creates offspring that are identical to the parent, and sexual reproduction does not. Now, just like before meiosis occurs in these cells in the ovaries and the testes, those cells do go through interphase as part of their cell cycle. And just as a reminder, question number 4 on the right-hand side of your notes asks what happens in the 3 steps of interphase. So we can fill in what takes place. During G1, the cell grows and matures. During the S stage, which stands for synthesis, the cell makes a copy of its DNA. And then during G2, the cell is going to get ready for division. Question number 4 also asks if interphase is considered part of cell division. Interphase is considered part of the cell cycle, which is the life of the cell as it grows and eventually divides, but it is not considered part of cell division. Interphase happens before cell division, and then cell division consists of mitosis, the division of the nucleus, and cytokinesis, the division of the cytoplasm and all the organelles within it. Okay, now meiosis uses the same cycle as mitosis, except meiosis is going to happen in 2 parts. So we see mitosis beginning in this cell cycle up here with prophase, metaphase, anaphase, and telophase. But if it was meiosis, you'd see prophase 1, metaphase 1, anaphase 1, and telophase 1, followed by prophase 2, metaphase 2, anaphase 2, and telophase 2. And prior to these 2 steps of meiosis, meiosis 1 and meiosis 2, we do have an interphase because that cell in the testes or the ovaries has to grow during G1, it has to copy its DNA, and it has to get ready for cell division. Okay. During meiosis 1, this is a very important step of meiosis, the number of chromosomes gets cut in half. So we end up going from 1 diploid parent cell, which in humans has 46 chromosomes, to 2 haploid daughter cells, which have 23 chromosomes each. And we're going to be breaking it down into the same stages of mitosis, but we're going to put a 1 after them to indicate that This is happening in meiosis 1, not meiosis 2. Okay. Prophase 1 in meiosis has similar events occur to the events that happen in prophase of mitosis. The DNA is going to coil up into chromosomes. We're going to start making the mitotic spindle, and both the nuclear membrane and the nucleolus are going to break down. There is one major difference from mitosis, however. During prophase 1 of meiosis, the homologous chromosomes are going to pair up. We haven't heard that term before. Homologous chromosomes are pairs of chromosomes in which you have one from your mom and one from your dad that have the same genes in the same places. Now, those might be different gene forms, like for example, the blue eye form versus the green eye form, but they're the same types of genes, like eye color, in the same places. Okay, so this is showing us a picture of what's happening during prophase 1, and we are going to label 4 things. We're going to label our pair of homologous chromosomes here, and then our pair of homologous chromosomes here. Notice that they've come next to each other. Whoops. We're also going to label these little fragments of the nuclear membrane that are shown in white. We're going to label this yellow spindle that's forming. And we're also going to label this right here. We haven't talked about this yet, but this is called crossing over. And it's when the homologous chromosomes come together and they exchange sections of DNA. So you see that the red part of this chromosome has now attached to the blue chromosome. And the blue part from this chromosome has attached to the red chromosome. That's crossing over, or the exchange of DNA between homologous chromosomes. Okay. And this is just another picture of this. Remember, you have one of your homologous chromosomes that came from your dad, so that's paternal, and one that came from your mother, so that's maternal. And we have 23 pairs of homologous chromosomes, or 46 chromosomes in the cell. All right, now, sometimes we say that the homologous chromosomes actually coming next to each other is called synapsis. And then when they do the actual exchange of DNA, like we see here, that's called crossing over. So lining up next to each other is synapsis, and exchanging DNA is crossing over.